Hello students, in the previous video we have seen what is cell cycle and what are the phases in the cell cycle. So cell cycle is a sequential or orderly manner of events that are taking place for the preparation of the cell to prepare its constituents, replicate its DNA and finally split the parent cell into two daughter cells. That is what is a cell cycle, and all the uh, all the phases in the cell cycle or transformation of one phase to the another phase is under genetic control, and it is mediated by the proteins called as cyclins. So one of the process of cell cycle is that is interphase, in which the cell preparation is going to take place for the cell division, and another phase of the interphase is cell division, or it could be M phase. Now the cell division is a final stage of cell cycle. It is when a cell divides to form daughter cells. A lot of reorganization occurs in various cellular components during the cell division, and therefore the cell division occurs in three different ways. Means after the cell has been produced its organelles, duplicated its DNA, means everything it has synthesized, then finally what happens is the division of the parent cell into two daughter cells. That is known as cell division, and which is the final stage of cell cycle. There are three types of cell divisions. One is mitosis, and another one is mitosis and meiosis. What is mitosis? Mitosis is also known as direct cell division, and this type of cell division, it is a mode of cell division in which a spindle fibers is not formed and chromosomes do not undergo any equatorial distribution. The nucleus and the cell cleave to form two daughter cells. There is a direct cleavage of the nucleus, and then the cell is going to divide the parent cell into two daughter cells. There is no additional disappearance or reappearance of nucleus and nucleolus or the formation of chromosomes, nothing. Just it is a direct cell division, direct cleavage of the nucleus into two daughter cells is being performed. Therefore mitosis is known as direct cell division, and it is going to take place in the cells of embryonic and fetal membranes. Ameiotic cell division is going to take place in the cartilage cells, okay, or in the meganucleus of paramecium. The cells are going to divide by amitosis. Another important type of cell division is mitosis, which is also known as indirect cell division. Why indirect? Here the nucleus is going to divide with the formation of the chromosomes, and then nucleolus and nuclear envelope. In the first stage it is going to disappear, and at the final stage it is going to reappear once again, and there are different types of complex mechanism is going to take place. Therefore it is known as indirect cell division, because in mitosis nuclear envelope first disappears, then chromosomes are distributed and nuclei are once again reformed, or around the nucleus once again the nuclear envelope is formed, and therefore mitosis is known as indirect cell division, and meiosis is a reductional cell division. Why? Even meiosis also can be considered as indirect cell division. There also the nuclear envelope first disappears, and later stages it is going to be reappeared, and the chromosome number is going to be reduced to half of the parental chromosome number, and therefore it is considered as a reductional cell division. Now in our study we are focusing on two types of cell divisions, that is mitosis and then meiosis, right? Mitosis and meiosis we are concentrating in our discussion today. Now what is this mitosis is also known as M phase, or it is known as dividing phase, or it is known as mitotic phase. Getting? Mitosis is also known as dividing phase, or it is a mitotic phase, or it is M phase. M phase is the phase that represents when the actual cell division occurs. When the actual cell division is going to occur, or it involves the separation of chromosome replication products and distributes the same equally into two daughter cells. Means whatever it is having the parental chromosome or parental genetic material, it is going to undergo, the cell will undergo the mitotic cell division and produces the two daughter cells which contain the same number and type of chromosomes as that of the parent cell or the mother cell. Getting? So, mitosis is also known as equational division. Mitotic division is also known as equational division. Why it is known as equational division? Because it contains the number of chromosomes as it is in the parent, and in the progeny cells it is going to remain same. It is going to remain same. As well as it is going to distribute the cell organelles which are present in the cytoplasm equally between the two daughter cells. Therefore, it is known as equational division. Why? In one case, it is going to exhibit the same parental chromosomes in the progeny cells also, and in another case, it is going to equally distribute the cell organelles between the two daughter cells. The cytoplasm and cell organelles are equally distributed between the two daughter cells. Therefore, it is known as equational division. Mitosis is also known as somatic cell division. It is also known as somatic cell division, as the cells, the somatic cells that form the body, will undergo the mitotic cell division, repeated mitotic cell division, in order to form the cell structures. So, it occurs during formation of body cells in case of both diploid and haploid in case of plant cells, but whereas only diploid cells in case of animal cells, and they are going to produce the cell structures. So, therefore, it is known as somatic cell division. It is known as somatic cell division. Somatic cells in the body are responsible for the growth. So, these somatic cells will repeatedly undergo mitotic cell division in order to produce the tissues and organs in the body. Therefore, mitosis is also known as somatic cell division, or it is also known as equational cell division. So, where all this mitosis is going to occur? In case of the animal cells, the single cell first formed during fertilization, that is zygote. This zygote is a diploid in nature, and now from this zygote the whole organism is going to be developed. Yes or no? Zygote is a first stage where when there is a male gamete sperm fuses with a female gamete ovum, it produces a zygote during fertilization. This zygote is single cell. Now from this small zygote a whole fetus is developed. From the fetus a offspring is developed. From the offspring adult is developed. Yes or no? So from this single zygote it undergoes repeated number of mitosis and cell differentiation to form the adult person or an adult individual, right? So it occurs in the human body or in the case of mammals from the zygotic stage, and it occurs in skin cells, bone marrow cells, in animal cells. Bone marrow cells will give rise to hematopoietic stem cells, and hematopoietic stem cells is going to form the lymphoid cells, progenitor cells. So all this, how they are produced? It is because of the mitosis. The cells are going to multiply because of mitosis. The number of cells is going to increase because of mitosis. So, in case of animal cells, in the diploid cells, the mitosis is going to take place mostly, and in case of plant cells, it occurs in meristematic cells. In plant cells, in meristematic cells such as stem cells, root tips, intercalary, and in the lateral meristems, the mitosis is going to take place. So, in the next year, in second year, you people will be studying performing an experiment, that is study of mitosis, which is important for your board exam, that practical. So there we are going to consider the root tips to study the mitosis. For the root tips to study the mitosis, we use a stain known as acetocarmine stain that you are going to study in the next year. So let us see in depth or in detail what is mitosis. Now we know what is mitosis. Mitosis is separation of chromosomes which were already replicated and distributing the same equally into two daughter cells. Now this mitosis is further divided into two stages. One is karyokinesis and another one is cytokinesis. It is further divided into two stages, karyokinesis and cytokinesis. Karyokinesis is also known as nuclear division or indirect division. Karyokinesis is also known as nuclear division or indirect division, and cytokinesis is known as cytoplasmic division. The division of the cytoplasm in order to separate two daughter cells. This is the final stage and that is known as cytokinesis. Karyokinesis, in which the nucleus is going to divide, but the cell is going to remain intact with two nuclei after the karyokinesis. Means the two daughter cells are not made independent, but they are together in the one cell. That is karyokinesis. Cytokinesis, you are going to separate the cytoplasm. So half of the cytoplasm carries one nucleus and another half carries another nucleus, resulting into two independent daughter cells. Karyokinesis is further divided into four stages. Karyokinesis is further divided into four stages. Stage 1 is prophase, stage 2 is metaphase, stage 3 is anaphase, and stage 4 is telophase. Karyokinesis is divided into four stages: prophase, metaphase, anaphase, and telophase. Now, each stage is not going to be differentiated. Getting? Each stage, it cannot be differentiated. There are no any particular boundaries from one stage to the another stage. All are just directed by the genetic control and because of the cyclin proteins. Because of the cyclin proteins that help from passage of one stage to another stage, and also based upon the specific events that is going to take place in each stage, the karyokinesis is divided into four stages. But there are no any clear cut boundaries that here the one stage will end and here another stage. It is all based upon the specific events that are taking place. Based on that, karyokinesis is divided into four stages: prophase, metaphase, anaphase, and telophase. Now let us see each stage or each step in detail. First, the prophase is going to be initiated soon after the interphase in the cell cycle. Okay, cell cycle constitutes interphase and M phase. So in interphase we have G1 phase, S phase, and G2 phase. Right? In G1 phase, the cell is going to prepare its proteins required for DNA replication, and in S phase DNA replication is going to take place, and in G2 phase RNA and other proteins are being synthesized. Spindle fibers, the proteins for spindle formation is also synthesized in the G2 phase. Clear? So after that, the cell is going to, after the G2 phase of interphase, the cell is going to enter into the M phase, and that is in the prophase. Now this prophase has two different phases. I mean early prophase and late prophase. Early prophase and late prophase based upon the events that are going to take place. So what happens in the early prophase? See now, after the replication of DNA in the S phase, the DNA molecules with the histone proteins are present in the form of intertwined threads. The DNA which is already synthesized, and in the G2 phase the proteins, in the S phase only the histone proteins and the DNA is replicated, right? Along with that, in the G2 phase RNA is synthesized. Now the DNA molecules, the histone proteins and RNA together, they are intertwined, means they are present in the form of coiled structure. That is how the chromatin is going to be present in the nucleus of the cell when it is not dividing. When it is not dividing, when the cell is in interphase, Okay, after the interphase, the DNA is replicated and it is present in the form of chromatin fibers or chromosomal material. Clear? Now the prophase initialization is marked by condensation of this chromatin material. The early prophase begins by condensation of this chromatin material, which is thin thread-like structures. Now this thin thread-like structures will uncoil and untangle. They are in the form of a coiled structure. Alright, now they are going to be uncoiled and untangled, and it is going to be thickening and shortening. That process is known as condensation to form chromosomes. It is to form chromosomes. That is going to takes place in the early prophase. After the prophase is begin. How we are going to now distinguish the prophase is, now the chromatin, whatever is synthesized in the S phase, now it is going to change its structure. From the chromatin fibers to it is going to be condensed into the chromosomes. Right? So that is the initialization of prophase. And next, in the S phase, not only the DNA is replicated, but also the centriole, the DNA is replicated in the nucleus, but the centrioles are duplicated in the S phase of interphase. Right? Now these centrioles will move towards opposite poles in case of animal cells. In animal cells only the centrioles are present and they move towards the opposite poles in case of animal cells, and from the centrioles rays known as astral rays are producing, that is resulting into asters. Means the centrioles along with the hair-like structure known as astral rays, these help in initiating the spindle fibers in case of animal cells. Okay, so this is, and spindle formation is initiated with the help of microtubules in the late prophase. In the early prophase only asters are formed, means asters with the movement of the centrioles to the opposite poles is going to take place in early prophase. But in case of late prophase, the spindle formation at the poles is going to begin in case of both plant cell as well as in case of animal cells. In case of animal cells, the spindle fibers are produced from asters. In case of plant cells, the spindle fibers are produced from the microtubules present in the cytoplasm. Clear? So, and apart from this, if you see the endoplasmic reticulum and Golgi complexes, okay, these are all disappeared in the prophase. In the late prophase, these are not observed under the microscope. As well as along with the Golgi complex and endoplasmic reticulum, nucleolus and the nuclear envelope is going to disintegrate and disappear. Now, however, the intact nucleus cannot be present throughout the cell division. It is going to disintegrate. If you see here in the diagram, early prophase and late prophase. In the early prophase, you can see the nuclear, you can see the boundary of this nucleus, that is nuclear envelope is present. Okay, with the help of the nucleolus is present. But in the late prophase, if you see, there is the disintegration of this nuclear envelope. There is a disintegration of this nuclear envelope and slowly it is going to disappear. And in further stages, this nucleolus and nuclear envelope will not be visible, as well as you cannot see the cell organelles within the nucleus during the cell division process. Clear? So, how we are going to differentiate early prophase and late prophase? In early prophase, chromatin fibers condense to form chromosome, and there is movement of centrioles which are previously duplicated in the S phase to the opposite poles. Okay, and in the late prophase, the spindle fibers are initiated from the centrioles in case of animal cells and from the microtubules in case of plant cells. And Golgi complexes, endoplasmic reticulum, nucleolus and nuclear envelope is going to disappear. That is going to mark the prophase. Once the chromosomes are almost formed in the late prophase, next phase is metaphase. Next phase it is going to enter is metaphase. So in metaphase, how you're going to identify the metaphase? So in your second year exams, you're going to identify particular stage under the microscope. Okay, so how you're going to identify the metaphase? Here the condensation of chromatin is completed. The disintegration of the nuclear envelope, which was initiated in the previous, in the prophase, is completed now. So there is no nuclear envelope. If you see the diagram here, transition to metaphase, you cannot see any nuclear envelope as it was in the prophase. So there is complete disintegration of the nuclear envelope that initiates the metaphase, and in this stage condensation of chromosomes is also completed, and chromosomes spread through the cytoplasm of the cell. Chromosomes are going to scattered in the cytoplasm of the cell. Now no more chromatin or thin thread like structures you are not going to observe, but you are going to observe as a chromosome. Now what is this each chromosome? Each chromosome contains its replicated copy known as chromatid. The two single threads of chromatin which is condensed, which is thickened and shortened, will form chromatid. So here the chromosome will contain chromatids. It is made up of two chromatids and which are held by the centromere, and the spindle fibers which was initiated in the previous phase, now they are going to attach to the chromosomes at a site known as kinetochore. The centromeres contain a disc-like structure known as kinetochore. There this, if you see here initially in the early metaphase stage, the chromosomes are scattered in the cytoplasm, and these are the spindle fibers which are initiated by the centriole and astral rays. They are going to be bind to the chromosomes. Can you see here from the opposite poles? So the chromosomes are made up of two sister chromatids which are held together by the centromere, and centromere contains a region known as kinetochore. To this region the spindle fibers are attached. Right? At this stage the spindle fibers attach to kinetochores of the chromosome, and these chromosomes are moved towards the equatorial plane. If you see the another diagram here, metaphase B diagram, now the scattered chromosomes in the cytoplasm are moved towards the equatorial plane. See here, which were scattered, they were dragged by the spindle fibers of the opposite poles to the equatorial region. Can you see here? To the equatorial plane, and this is known as congression. The term used is known as means bringing of these chromosomes towards the equatorial region by the spindle fibers is known as congression. Now the arrangement of chromosomes is such a way that one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole, and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. Can you see? Now this is one chromosome here. This is containing two chromatids. Now each chromatid is connected to the spindle fibers of the opposite pole. One sister chromatid. Now these two chromatids which are identical for the same chromosome are known as sister chromatid, and each chromatid is connected to the spindle fiber, and they are arranged in the equatorial plane. They are arranged in the equatorial region. So the plane of alignment of chromosomes with their centromeres on the equator is known as metaphase plate. It is known as metaphase plate or equatorial plate. What is this equatorial plate or metaphase? Okay, each chromosome is aligned with their centromeres at the equatorial region. So all the chromosomes, when they are aligned at the in the metaphase at the equatorial region, is known as metaphase plate, or it is known as equatorial plate. It is known as equatorial plate. So this marks the completion of the metaphase, and metaphase is the best phase to study the morphology of the chromosomes, number of chromosomes, everything you can study in the metaphase stage. The number, because in the metaphase stage the chromosomes are aligned at the equatorial region, and all the studies of the chromosomes is possible to study in the metaphase stage of the cell division. Moving on to the next phase, that is anaphase. Next stage is anaphase. So what happens in anaphase? Now in the metaphase, what is happening? The spindle fibers are attached to the kinetochores of the centromeres of a chromosome. In the metaphase, now finally what should happen? Ultimate goal of the mitosis is the nucleus has to be divided. Means the nucleus containing the genetic material in the form of chromatin has to be divided. So these chromatin now have condensed in the prophase. Condensation began in the prophase, continued to metaphase, and completed in the metaphase, and the complete chromosomes are formed in metaphase, and these chromosomes must be distributed into two halves. Okay, because they are duplicated chromosomes, and they have to segregate it, and one set of chromosomes must be created. So in anaphase, what happens? The spindle fibers split the centromere which is attached at the kinetochore and makes these daughter chromatids to move to the opposite poles. So the centromere of each chromosome arranged at the metaphase plate splits by the spindle fibers. Okay, and it is going to move these daughter chromatids to the opposite poles. Moves this daughter chromatids to the opposite poles, and the movement of chromatids to the opposite poles is known as anaphasic movement. Both the chromatids move towards opposite poles, which is known as anaphasic movement. What happens? The centromere of each chromosome arranged at the metaphase plate is split by the spindle fibers, and the chromatids are going to be moved towards the each. Now there are two sister chromatids for each chromosome, so each chromatid will be moved to the opposite pole. Each sister chromatid will be moved to the opposite poles, which is known as anaphasic movement. As the chromatids move from equatorial plane towards the poles, the centromere will be at leading edge. Getting? When they are moving towards the opposite pole, the centromere, see, these are centrioles present here, and these are astral rays, and these are the spindle fibers. Okay, when the chromosomes were at the equatorial region, now these chromosomes, the centromere was split, and the chromatids are pulled towards the opposite. These two together was one chromosome. These two are each sister chromatids, and each sister chromatid is pulled towards the opposite pole. Okay, and if you see this, as the chromatids are moving towards the away from the equatorial plane towards the poles, the centromere will be at leading edge. Means it is toward, this is the centromere, the small circular part here is the centromere. So the centromere is moving towards the pole, so it is considered as a leading edge. And the arms, in the Cell the Unit of Life chapter, you might have studied the chromosome structure. So the chromosomes will have two arms. These arms are going short arm and long arm. These arms are trailing behind. So this is leading. Centromere is leading, and the arms are trailing behind pole, and the arms of the chromosome, which is going to be trailing behind the behind and reaching towards the opposite poles. Right? Here, the chromosomes are visible during the anaphasic movement, such as it is going to exhibit different shapes of the chromosomes are visible. V shape, J shape, I shape, or L shape are visible during the anaphasic movement. So V shape means it is going to denote metacentric chromosomes. You have studied, right? Different types of chromosomes: metacentric, submetacentric, acrocentric, and telocentric chromosomes. So V-shaped chromosomes are metacentric chromosomes, L-shaped chromosomes are submetacentric chromosomes, J-shaped chromosomes are acrocentric chromosomes, and I-shaped chromosomes are telocentric chromosomes. This is visible during the anaphasic movement in the anaphase stage. So the anaphase stage you are going to mark by the splitting of the centromere and movement of the sister chromatids towards the opposite poles. Next stage is telophase. This is the last stage of the mitosis. That is, in the mitosis, karyokinesis is there. So the karyokinesis is divided into prophase, metaphase, anaphase and telophase. And telophase is going to remain as the last phase of karyokinesis. Okay, means which is going to form the complete nuclei, the parent nucleus which was divided into two daughter nuclei. So that you can mark the telophase as chromosomes that are moved to the opposite poles, that is a set of chromatids, each set of chromatids that are moved towards the opposite poles. Now they are not going to remain in the form of the chromosomes. Now these chromosomes are decondensed back, they are getting decondensed, and it is once again present in the form of chromatin, a thin network like thread like structures. Okay, once again they are coiled, okay, and they are elongated and coiled, which was thick and short as chromosomes. Now these are going to be elongated and they are once again coiled, and they are going to form the mass of group or cluster of thread like structure, and it is going to form the chromatin. So chromosomes which are present in two different groups will lose their identity as a distinct chromosome, and therefore they are going to form a network of chromatin at the opposite poles. And what happened, once again the nucleus is going to be complete in case of eukaryotes when there is a nuclear envelope present. So the nuclear envelope is going to be formed around this chromatin materials at the opposite poles. You can see here the nuclear envelope is formed around the chromatin material and at the opposite poles, and the nucleolus will reappear once again. Okay, and the Golgi complex, endoplasmic reticulum and other organelles which are duplicated and which were reappeared in the prophase will once again disappeared in the prophase will once again reappear in the telophase. Okay, all this together will result into formation of two daughter nuclei. So there is here one daughter nuclei and this is one daughter nuclei. Initially there was only one single nucleus at the beginning of the prophase, but at the end of the telophase two daughter nuclei are formed which contain each a set of chromatin material, that is chromosomes, as that of the, or it is identical to the parental chromosomes. So therefore it is known as equational division, and telophase is considered as reverse prophase. Telophase is considered as a reverse prophase. Why it is considered as a reverse prophase? Because it is exactly the events that are taking place in telophase are against the prophase. In case of the prophase, the chromatin material is undergoing condensation. In case of telophase, the chromosomes are decondensed to form the chromatin, and there the nucleolus and nuclear envelope is disappearing. Here the nucleolus and nuclear envelope is reappearing. There the Golgi complex and endoplasmic reticulum was not seen. In case of telophase, the Golgi complex and endoplasmic reticulum along with other organelles are observed. So therefore telophase is considered as reverse of prophase, and this is going to end the karyokinesis, that is a division of nucleus is completed. Right students? Now the division of nucleus is completed, but still the two cells are not separated independently. Therefore once again the cell has to undergo another stage of division known as cytokinesis. Because the mitosis is divided into two stages, karyokinesis, that is division of nucleus, and cytokinesis is division of cytoplasm. Though the cell is containing two nuclei containing the same amount of genetic material in both the cells, both the nuclei, but the nuclei is not segregated into separate cell. So cytokinesis is the division of cytoplasm of parent cell into two daughter cells carrying equal part of the cytoplasm, resulting in formation of two daughter cells, which marks the completion of the cell division. Getting means whatever the cell organelles that are synthesized and present in the cytoplasm must also be distributed equally to both the cells, and they should be made independent. That is done by the process known as cytokinesis. So cytokinesis is furthermore divided, or it is having two types of cytokinesis. One is known as cell furrow method, another one is cell plate method. Cell furrow method is a type of cytokinesis that is going to take place in animal cells. Soon after the mitosis, the animal cells will undergo cell furrow. Soon after the karyokinesis, the animal cells will undergo cell furrow method. But in case of plant cells, soon after the karyokinesis, the plant cells undergo cell plate method. What is this cell furrow method and what is this cell plate method? Let us see what is cell furrow method. See, if you see the cell in case of anaphase. So what happens in case of anaphase, how the cell is resembling? The round cell, how it was in prophase, now it is bit elongated in case of anaphase. See, in the metaphase, the cell was round. When you came to anaphase, the cell is the cell membrane. Now whatever the pictures you are seeing here, the cell division, it is for animal cell. So animal cell do not contain cell wall, but they contain cell membrane. So the nuclear envelope is been disintegrated, so only the boundary which is present here is the cell membrane. So this is the cell membrane, right? Now this cell membrane becomes flexible during anaphase. That is the reason it has been changed from the circular form to the elongated form. And in the next phase, that is in the telophase, it forms a constriction or an invagination or a furrow. See here, it is forming a constriction or an invagination known as furrow. How this furrow is formed? So it is formed by the method known as cell furrow method. Now this cell furrow method, it appears in the telophase in the cell. A furrow is appeared in the telophase. How this furrow is formed? How this furrow is formed? It is formed due to the spindle fibers that are present in the equatorial region. Now you have seen in metaphase, when the chromosomes are aligned, the spindle fibers are present along with the chromosomes in the equatorial region. Now these spindle fibers, okay, they are going to be condensed, okay, dense and condensed, and forms a structure in the equatorial region, and along with that the microfilaments are also formed at peripheral below the cell membrane in equatorial region. If you see here, so at the equatorial region there were spindle fibers, okay. These spindle fibers are densed at the equatorial region, and along with this the microfilaments are present, okay. All this together is going to contract the cell membrane. It is going to contract the cell membrane, and therefore a furrow or an invagination is being formed in the cell. So microfilaments formed at the peripheral below cell membrane in equatorial region, and the proteins cause contraction of these peripheral filaments, which is leading to the formation of this constriction or invagination, or it is known as a furrow. Now what happens, this furrow, whatever the furrow that is formed is going to be gradually increase its size towards the center, or it grows towards the centripetally. It is going to grow towards the centripetal region, means towards the center, that is going to deepen towards the center, and this furrow is going to ultimately join in the center and divide the cell cytoplasm into two cells. Now this invagination slowly it moves towards the center. what will happen? It is going to form two daughter cells. It is going to form two daughter cells. So the cleavage furrow is formed with the help of proteins, that is actin and myosin, which are present in the cytoplasm, okay, and the microfilaments involved here, and the spindle region is formed at the center. All together is going to form the cell furrow, and this cell furrow is being deepened gradually as it moves toward the center, and once it is deepened completely, it is going to divide the two cells. It is going to divide a single cell into two cells which contain two sets of nucleus, which is going to contain two sets of nucleus. Sorry for the interruption. It is going to produce two sets of nucleus, and therefore it is resulting into two daughter cells. The mitosis is going to result into two daughter cells, each containing the same amount of genetic material. Next is cell plate method. Now in case of plant cells, it contains the cell plate. In case of plant cells, it is going to contain the cell plate. Cell wall is present in case of plant cells. So the division of this cell wall between the two daughter cells is known as cell plate method. So equatorial part, similarly as that happened in case of the animal cells, in case of plants also, in the equatorial region the spindle fibers are going to enlarge and they are going to combine with the microtubules, and it is going to resulting into a structure known as phragmoplast. It is going to form a structure known as phragmoplast at the center. So what is the phragmoplast? Act as a precursor for the formation of the cell plate. It forms a precursor to form the cell plate, and this phragmoplast that is formed during the telophase is going to carry the polysaccharide vesicles that are being produced by the Golgi apparatus. Now you know what is the function of the Golgi apparatus. It is going to produce the secretory vesicles, and these secretory vesicles are carried by the phragmoplast for the construction of the cell plate. Another structure known as cell plate is being produced by the formation of the phragmoplast, as it is going to transfer the polysaccharides required for the construction of the cell wall. We know that all the plant cell walls are made up of most of the polysaccharides. So these cell plate whichever is formed, which is a thin layer, now this is going to grow towards the outside. Whereas in case of animal cells, the furrow was formed at the periphery and it was deepening towards the inside or towards the center. The cell plate is formed in the center and it is growing towards the outsides, okay, at peripheral region, and resulting into two cells. It is going to form two daughter cells. So later the cell plate is solidified, okay, with the calcium pectate, and which is known as middle lamellae. The cell plate is going to act as middle lamellae. What is middle lamellae? You have studied in the cell unit of life chapter when you are studying the structure of the cell wall. It is the outermost layer of the cell wall which acts as a glue or cementing agent between two adjacent cells. So first the cell plate is going to be formed, and later from this middle lamellae or the cell plate, the furthermore layers of the cell wall is going to form. The furthermore layers are the primary wall, secondary wall, tertiary wall. Slowly it is being formed, okay. So the initial segregation of the parent cell into two daughter cells is by the formation of the cell plate. To form the cell plate, a precursor known as phragmoplast is formed, and this phragmoplast is formed from spindle fibers at the equatorial region and with the help of the microtubules, whose function is to carry the polysaccharides produced by the Golgi body in the construction of the thin layer at the equatorial region, that is cell plate, which grows peripherally or centrifugally or towards the peripheral regions and extends a new layer known as middle lamellae, which acts as a primary wall segregation between these two cells. Lastly, we are going to see another concept known as syncytium cells. What are these syncytium cells? Now, if in some cells the nuclear division is going to take place, that is known as karyokinesis, but in some cases if this karyokinesis is not followed by the cytokinesis, means the two nuclei are produced within the cell, but if there is no cytoplasm division, that is no cytokinesis is going to take place, that resulting into a multinucleated condition which is known as syncytium. So under the conditions like the nucleus divide but the cytoplasm does not, it produces a syncytium cells, or several cells merge together retaining their nuclei but the cell membranes or the cell walls are being broken. In that condition also it results in the syncytium cells. Clear, students? So the syncytium cell examples is, it is present in the skeletal muscles of human beings, they are present in the embryonic stages of the pregnancy in women, and it is present in other mammalian cells. So syncytium cells are made up of embryonic cells and the placental cells. All contain these syncytium cells, means multinucleated cells. It could be either there is no cytokinesis taking place, or after the cells are formed, okay, they merge together cleaving their cell membranes but retaining their nuclear envelope, that is also going to form the syncytium cells.